Well, welcome everybody to the fourth session of the 2026 Spring Series on Magnetic Fields in Biology. I'm Geoffrey Guy, the chairman and founder of the foundation. And today we welcome Betony Adams, our own Betony Adams, to talk a little bit and then introduce our speaker, Massimo Maffei from Italy. I think we're going to find this absolutely fascinating, looking at magnetic fields in plants. So without further ado, Bethany, can we open the meeting to you?
Thank you so much, Geoffrey. It gives me great pleasure to introduce this fourth session of the spring series on magnetic fields in biology, where Professor Massimo Maffei will be presenting his work on magnetic field effects in plants. As a quick recap, why are magnetic fields interesting in the context of biological systems? Well, all life on Earth evolved in the Earth's magnetic field. At present, the strength of the geomagnetic field ranges from around 30 microteslas to 70 microtesla. It has also been subject to some flips in direction over the history of the Earth, and has some areas in which it diverges from the latitudinal pattern, such as the South Atlantic anomaly. How do magnetic fields interact with biological systems? Well, there are various ways or theories that have been put forward, either through magnetic materials such as magnetite or through spin chemistry, where the radical pair mechanism describes how magnetic fields can influence chemical reactions. And we heard a bit about this in the first session of the series, which is very much worth a watch if you missed that. It is also maybe useful to have some idea of what is meant by a weak magnetic field, which you will often hear the Earth's magnetic field described as. For some perspective, MRI technologies used in medical imaging use strong fields of the order of teslas. The Earth's field is weak in comparison to this, measured in microteslas, which is roughly 6 orders of magnitude weaker. But hypermagnetic fields can get even weaker, ranging from no fields altogether to fields of nanotesla strength, which becomes relevant when we start to look at environments outside of Earth, such as Mars. So in session 3, last session, we heard Dr. Sanika Gael and Professor Wendy Bean tell us about the evidence for magnetic field effects in animals, while spin chemistry and quantum biology has mostly focused on behavioral effects, such as magnetoreception. There's growing evidence that magnetic fields may have profound effects on cellular function. And for those of you who missed last session, I also very much recommend watching that. The fourth session is part 2 of this focus, building on animal experiments by extending the data to other biological systems, such as plants. Experimental evidence suggests that magnetic fields may influence a range of plant processes, including seed dormancy and germination, differential growth patterns, photosynthesis, overall robustness to stress. And this magnetic sensitivity has various implications, from improving agricultural productivity and resilience to informing medical research, and even shaping our understanding of how organisms might adapt to the altered conditions encountered in space. But I will leave it to today's speaker to tell us the interesting details. Professor Massimo Maffei is Professor of Plant Physiology in the Department of Life Sciences at the University of Turin in Italy. He is coordinator of the Ministerial Centre of Excellence for Biosensing. He's head of Department of Plant Biology and Deputy Dean of Faculty of Sciences. He's editor-in-chief of the International Journal of Plant Interactions, and he's a member of the advisory board of the Journal of Essential Oil Research. And he is reviewer for several international journals dealing with plant physiology, phytochemistry, natural product chemistry, and plant biology. And his research interests and activity is focused on plant interactions with the surrounding biotic and abiotic environment, with particular reference to signal transduction pathways, gene expression, and metabolomics. And without wasting much more time, let me hand over to Massimo.
First of all, thanks a lot for giving me the possibility to show what plants are doing when the magnetic field is changing. As Betony already said, it is interesting to see how the magnetic field is changing in our planet. So the magnitude is the first thing that we can observe. As Betony said, there are differences in our planet in the intensity of the magnetic field. Here is the what is called the South Atlantic anomaly. It's an anomaly because the values of the geomagnetic field are very low. There are about 20,000 nanotesla or 20 microtesla. And you can see also these areas where the geomagnetic field is higher. We are in this area in Europe where it ranges between 40 and 45 microtesla. Of course, this is not a constant situation because there are reversals. So every 100,000 years, there is a reversal in the geomagnetic fields. So there is a swap between the south and north. And during this reversal, there is this kind of situation. And this is important because the geomagnetic field is protecting us from solar wind and cosmic radiation. So the effect is to protect the biosphere. And we are developing life, and life evolved in the presence of this shield. So we are continuously protected by the geomagnetic field. So during reversal, this shield is really, really reduced. And so this enhances the probability that cosmic rays and many other kind of radiation can reach the biosphere. So this is in general what we know about the effect of magnetic field on plants. So I'm working mainly on near null magnetic field effects, and I will deep my presentation on this field. But there are also several papers. Maybe most of the literature is based on increased magnetic field. So sometimes it is called hypomagnetic field, but actually it is still in the millitesla range. So up to the, I mean, if you go over the geomagnetic field, so values higher than 45 or 50 microtesla are not low or hypo, but they are hypermagnetic field with respect to geomagnetic field. Nevertheless, there are several papers that deal with the increased magnetic field and their effects on plants, and in general there is root stimulation and stimulatory effects on productivity. The mechanism is still unknown. It involves hormonal and molecular responses. So there are receptors that might be involved, like for example the cryptochrome, even though it is not so clear the involvement of cryptochrome as it is in animal. And of course, auxin, gibberellins, and many other hormones are altered by the fluctuation in the geomagnetic field or in the alteration of the geomagnetic field. In terms of global agricultural and economic impact, of course, if we can correlate the yield with the presence of the magnetic field, then we can prepare for strategies to increase productivity, to have economic outcomes. And of course, we can also manage the magnetic field technology in order to have crop enhancement and to have eco-friendly, affordable, and non-toxic residue or radiation. In my field, I deal with very low magnetic field, like I would say near null magnetic field, because I'm involved in several projects in astrobiology, financed by the National Space Agency. And so it is important for me not to understand what it will be the response to higher magnetic field, but to understand what be the fate of life in general and in particular of plants when we will have to move to Mars or Moon where there is no magnetic field at all. And also there is a different gravity. So in our studies, we try to combine the effect of near null magnetic field with hypogravity, or let's say mimicking the gravity of these planets. So this is also interesting because in our solar system, we do not have only this kind of situation of low magnetic field, but we have also, for example, moons like Europa or Enceladus, which are affected by the magnetic field of their planets. And in this case, we have higher magnetic field with respect to Earth. So it is really interesting to understand how living organisms interact with these different environmental situation in terms of magnetic field. Our research, as I said, is based on discovering what is going on outside our planet. As I said in my short introduction, we are protected by our biosphere, so we are unable to survive to what is outside this biosphere. And if you want to adapt to these different environments, it's important to understand what it will be the fate of plants or other animals when they will be transposed to other systems where no magnetic field or different gravity situation occur. Our focus on research, of course, is to try to isolate those traits that can tell us how we can improve the stress tolerance in plants. And for this, we use genomics and metabolomics approaches. We want to deepen the mechanism of magnetoreception in order to create plants that will adapt, that will show mechanism of acclimation that are good enough to sustain life in other systems. Of course, nobody thinks of growing plants directly on the Mars soil or Moon soil exposed to the environment, because this would be not possible. But even if we will build space greenhouses, these space greenhouses will face the actual situation of the planet. So no magnetic field, low gravity, and so on. So what we do to study this particular environment that we want to build around the plant. So we use this triaxial Helmholtz coil system, which is a very, very finely calibrated system that allows us to understand exactly the magnetic field that we are removing from the site of plant growth. Actually, we are just taking out the geomagnetic field and reproducing a near null magnetic field environment. In combination with this system, we have a random positioning machine that allow us to simulate gravity situation in different planets, or simulate the microgravity there is in the deep space or International Space Station, whatever. Anyway, we can combine the effect of no magnetic field with low gravity. In this case, we were simulated the Martian gravity at 0.38 g, and we were also simulating the Mars magnetic field. So we can combine these things. How we control the system? So this is the real size of our system. So it is big enough to allow plant to grow and also to go from seed to seed. Of course, it depends on the plant. But we finally control the system by continuously monitoring the magnetic field values. So we have a flux gate that is positioned at the real center of the system. It is controlled by different, of course, the coils are controlled by power supplies, and then we have the magnetometer here, computer, power resistance, and system to standardize the current. But what we did is to develop also softwares that allows us to control in real time the situation of the magnetic field. This is the magnetic field, how it is actually changing in our planet. So there is no way to have a constant fixed magnetic field, because the magnetic field continuously changes because of the geodynamo of our planet. But what we can do is to confine the magnetic field values in a range. This is between 100 milli, so 100 nano to 0 nanotesla. So we expose plant to conditions where we have an average value, in this case was about 49 nanotesla, and we can be sure that we have we keep tracking the values, but we also developed what is called the twin the twin system for the control of this instrument. So we've been working in collaboration with the National Research Center Institute in Milan. They are specialized in electromagnetic fields. And so they developed this digital twin system that allows us to understand exactly the density of the magnetic field that we are reducing. So as you can see, from this center to about 35 centimeters from the center, we are quite sure that we have about between 20 and 40 nanotesla. And then as soon as you move toward the edges of the coils, then you get from 10 to 45 microtesla, which is the actual value of our laboratories. So in this way, we are quite sure that when the plant grows, we know exactly when the plant will be exposed to what we call the near null magnetic field and when it goes out, so it goes out of the control of the magnetic field. These are our labs. So we can do several things from metabolomics to genomics. We have confocal microscopes. We have PCR machine and liquid and gas chromatography. And these allow us to go straight from this environment to the lab and analyze what we are doing. And these are the labs where we develop most of our research. So let me tell you about the beginning of all of this history. So I met this guy, Angelo De Santis. He's an expert in paleomagnetics field. So he is from the National Institute for Volcanology and Geophysics in Rome. And he provided me with this picture. He said, these are the reversal and the normal situation of the geomagnetic field. What can we do with this? So I said, well, let's play this game. So let's put when species and families developed and see whether they combine with your graph. So we played this game and we published this game in Trends in Plant Science in 2014. And every time that there was a normal magnetic field, the, let's say, paleontological or geological reconstruction of the species in the angiosperm were always matching the normal situation. There was nothing going on in the reverse. Of course, this was some sort of a game, but last time we were talking about this, and so I put this slide, which tells you in the bottom, you can see the normal and reverse as black normal and white reverse magnetic field. And here you can see the evolution of primates, humans diverging from chimps and gorillas. And you can see that there are also in this case some very nice correlation, like in this case the Ardopithecus that is disappearing every time that there is a normal, or some other one that are overlapping some periods. But the most interesting thing is that the Homo sapiens never experienced a reversal. So we evolved in a non-magnetic field, and this very thin line is when the Neanderthal disappeared, and is the last 2,000-3,000 years of continuity of reverse magnetic field. So besides of this, so we had to demonstrate that this was true. So we used our system, and in collaboration with Chris Rogers, at that time it was in the University of Oxford, now he's in Cambridge. And so we reversed with our system the geomagnetic field. And what happened is that in the reversal, plants were behaving differently. So indeed, if you reverse the geomagnetic field, you have at least a phenotypic effect. We were checking some genes in order to see whether there was an effect, and indeed, shoots and roots were behaving differently, and all were reacting to the reversal of the magnetic field. These are gene expression where you express reverse against normal magnetic field. So indeed, plants are able to perceive not only intensity, but also orientation of the magnetic field. And this was quite interesting to notice. But we need to know more about that. So in that period, there was a group in China, the group from the Chinese, let me remember. Anyway, Professor Xu was one of these researchers, Chinese Academy of Science, and Professor Xu was supposing that the reduction of the geomagnetic field was delaying flowering time. It was interesting, so we indeed did the same experiment, but we extended the experiment to genomics as well. But what we did was to grow plants in, let's say, in generation experiments. So plants were growing in a geomagnetic field, and this is the timing of flowering for Arabidopsis in normal conditions. But then when we move the plants and sow the plants in near null magnetic field, there was a flowering delay. We took the seeds inside the machine and then we sow again, and we got 2 generations. And as you can see, there was still a delay in flowering. It was maintained after the 2nd generation. So we thought it was something epigenetic going on. But then when we took the seeds of the last generation, we put in normal magnetic field, the phenotype was completely normal. So the epigenetics, it happens, is not inherited. It happens during the exposure to near null magnetic field, but it's not inherited. Anyway, the chronic exposure of plants causes, of course, some difference in leaf area index and also in stem length. But it was interesting to understand how it was possible that this change in the magnetic field was affecting of course, the flowering time. So we explored all the genes that are involved in the flowering. So the clock genes, the photoperiod genes, the vernalization genes, and also those of the gibberellin pathway, which is controlling part of this mechanism. Gibberellins are hormones that are involved in flowering. And then the regulatory network, both in leaves, but also in the floral meristem. We did this study in early, intermediate, and late times. And what we could observe that there is a strong downregulation of most of the genes already in early times of plant development. So there is something that is perceived by the plant. It's some sort of magnetoreception, and this has profound effects on those genes that are controlling flowering. So we need to understand whether this flowering delay was also connected to what is the, let's say, the engine of controlling flowering, which are the photoreceptors. So we start this collaboration with John Christie. He's from the University of Glasgow. He's an expert of cryptochrome. And we did an experiment mimicking the same experiment that we did, but this time with Cry1, Cry2 mutants. Cry1, Cry2 mutants should completely delete the cryptochrome response. But as you can see, we got the same flowering delay in plants, Cry1, Cry2 in GMF or in NNMF, so in near null magnetic field. Meaning that probably cryptochrome is not the only one responsive of the changes in magnetic field as it is known in animals. So this experiment was quite simple. So we used darkness in scotomorphogenesis in order to understand whether plants were responding differently to geomagnetic field or near null magnetic field. And from the range of the experimental duration, we saw no change in the morphological results. But what happens when you use blue light in normal condition? Usually cryptochrome 1 is makes phosphorylation, but cryptochrome Cry2 is degraded. What happens? Nothing happens to phytochrome A and B. And for phototropin, blue light activates and phosphorylates the phototropin protein. In red light, in normal condition, Cry1 and Cry2 interact with phytochrome, and so phytochrome is degraded, and Phi A and Phi B are degraded in this way. What happens when you take out the geomagnetic field? No more phosphorylation in Cry1, and Cry2 is not degraded. It's still, you have phosphorylation in phototropin 1. What happens with the red light is that if you take out the geomagnetic field, you don't have any more the degradation of phytochrome B, and you have only the effect on phytochrome A degradation, the effect from Cry1, Cry2, and Phot1. So the results is that there is a cooperation between photoreceptors, no matter the wavelength that you are using. Also, these results were interesting because we could demonstrate there is a light-independent mechanism of magnetoreception in plants. Of course, when you talk about plants and you see difference in growing habitudes or growing performances, you think that probably there is something going on in the perception of the magnetic field and the mineral nutrition. So we did these experiments. So we've been using gene microarray and also capillary electrophoresis to do ionomics analysis on cations and anions. And what we found is that the geomagnetic field is important for mineral nutrition. When you take out the geomagnetic field and you go to near null magnetic field, you get significant changes both in the content of the ions that are absorbed by the roots, but also in the channel and transporter activity. So we could see, for example, that if you have a time course experiment from 10 minutes to 96 hours, you see changes, for example, in root and shoot absorption of mineral nutrients like nitrate or phosphate. The same was for potassium or for magnesium. And also we could see, a significant difference in the gene expression of, let's say, transporters and channels. When you talk about this kind of exchange between the soil and the root, what it mediates everything is the plasma membrane. So we were curious to understand whether the geomagnetic field is an important player in lipidomics. So what we did was to once again have a time course experiment from rosette to SITSAT, and to understand whether there were changes in the lipid composition. So we analyzed two kinds of lipids, the surface alkanes and the fatty acids. Of course, fatty acids are important because are constituent of plasma membranes and all other membranes in the plant. What we saw was, even in this case, we did a microarray analysis, and we found that there was a striking difference in the surface alkanes in the leaf surface, and so we could see that there was an increase in these surface alkanes along with the plant development, but also we could check the gene expression of major genes involved in the lipid metabolism, for example, a series of ketoacyl synthesis, but also lipid transfer protein, and we could see that if you reduce the geomagnetic field, then you have an increase in the activity of these genes. So indeed, the geomagnetic field is also playing a role in the normal lipid metabolism. But we need to confirm one of our hypotheses is that the roots and the shoots are differently responding to the geomagnetic field. So with this experiment, that was done in collaboration with Ivan Paponov from Aarhus University and Judith Fligman from, at that time, she was in France, in Nantes. And what we did in this experiment was to have a time course, huge microarray experiment. We got about 64 different microarrays analysis, and we had to understand whether roots and shoots were displaying the same response or were differently responding to the alteration of the geomagnetic field. So what we did was just to run these microarray studies from 10 minutes to 96 hours. Once again, we want to focus on early events in the geomagnetic field perception, and we found that 49% of the genes were reversely regulated between shoots and roots. And also that was a biphasic response, and sometimes there was some hormetic response, something that we also noticed to happen in several other experiments. So in general, this is what happens. If you take this microarray experiment and you see roots and shoots from early to late time, you see that there are some sort of different pattern. So these genes are downregulated in the roots, but are upregulated in the shoots, and here is the reversal. And so these make us think that there are probably different timing in magnetoreception, or different organ possess different way of perceiving the magnetic field. So I was presenting these results. They were preliminary results at the Kennedy Space Center several years ago. And one of the questions was, well, you should also not only have time course, but also have magnetic field changes in order to see whether there are some correlation between the intensity and the timing of magnetic field exposure. And we did it. So as you remember, I said that the geomagnetic field has different places where the geomagnetic field is lower. So for example, we could imagine that if you sample plants here in the north, could be different than sampling plants in the Central America or Central Africa. So we did this kind of experiment. I'm just showing few of these genes, but we were mimicking changes in the geomagnetic field intensity from 40 nanotesla to 60 microtesla. And as you can see, there are some genes that in roots and shoots are behaving differently according to the magnetic field. Sometimes you have completely different patterns, as in this case, or reverse patterns, or similar patterns. But what it could demonstrate this experiment is that indeed, if we have, let's say, a multi-center study that involves several institutes in several parts of the world with the same plant in the same condition, we will get this kind of situation, because we are moving between, let's say, 18, which is the anomaly in the Central Atlantic, to Anchorage that has about 60 microtesla, and we should be able to see this kind of effects. This reflects on the ecotypes. We are running an experiment in these days with different Arabidopsis ecotypes, and we see that every ecotype has evolved in a particular magnetic field, and it responds to different magnetic field exposure. So this is also relevant in terms of adaptation of plants in our changing magnetic field environment. So coming back to cryptochrome, we know exactly that cryptochrome is involved in magnetoreception. Cryptochrome goes from the ground state to a conformational change that is depending on the light, and this activates the cryptochrome with phosphorylation. The activated state of cryptochrome can also be changed during the night period with what is called the dark reoxidation period. During this dark reoxidation period, molecular oxygen is involved, and if you apply different magnetic field during this period, you get different responses. So Margaret Ahmad from Paris University and Sorbonne published this nice paper that I readapted to make this image. So actually what happens is that when you have the phosphorylation of the krypton, you have the activated state during the night, and during magnetic field changes, you have this interaction with the oxygen, and you have production of reactive oxygen species, and then you have the coming back of the reaction that goes to the normal situation. So this makes you think that there is something that is very common in birds. We know that birds use cryptochrome for orientation, but probably you do not know that 70% of migratory birds fly at night. So they cannot use light in order to manage the cryptochrome. So definitely they have to rely on this dark reoxidation period after they have activated cryptochrome during the day light flight. So based on this consideration, and due to the importance of the oxygen, we tested whether reactive oxygen species could be involved in the plant responses to near null magnetic field exposure. So what we did was once again to have microarray studies on Arabidopsis thaliana, and this time we combined the gene expression, and we focused gene expression of ROS scavenging and producing enzyme. The bold phase one are the producing ROS, and the normal written genes are the ones that are scavenging the enzyme, the ROS. So what you can see is that you have in a time course differential expression of these genes when you expose to near null magnetic field. And once again, you can see that roots and shoots behave in a different way when you analyze the gene expression of these ROS scavenging genes. Parallelly, we were also testing the ability of plants to interfere with ROS. As you know, plants are producer of antioxidants, and most of the antioxidants are in the range of polyphenols. So we analyzed and we did the full metabolomics of the polyphenols, and we also tried to correlate the gene expression of the genes that are involved in polyphenol production, and we could see clearly that there was a correlation between the change in the, let's say, in the ROS perception of the plants and the polyphenol production. So indeed, what we could demonstrate is that if you expose plant to reduce magnetic field, you have a reduction in the ROS production. This reduction in the ROS production is something very weird, because if we consider that the magnetic field is inducing some sort of stress, usually during stress you have an increase in ROS, not a decrease in ROS. And this decrease in ROS was also accompanied by the decrease in polyphenols. So at the end, what happens is that if you expose plant to reduce magnetic field, then you reduce the ROS production, but also the ROS power of plant to detoxify these compounds. Coming back to the magnetoreception, in 2016 there was this nice paper published by a Chinese group in Nature Materials, where they were proposing a mechanism, a potential mechanism for magnetoreception. This mechanism was a combination of cryptochrome and also iron-sulfur cluster assemblies. Iron-sulfur clusters are important because they are widespread in the plant, but also in general in all organisms. They are very much conserved from bacteria to humans, and so and these clusters are important because, according to this paper and to the results that they got, they can form some sort of a complex where there are photoreceptors around the core that is made by these iron sulfur clusters. So this system, which has been called MEGAR, allows organisms to detect not only the intensity, but also the orientation of the magnetic field. This has been proved in several organisms, from bacteria to pigeons to butterflies, and even humans have the same gene, but if, of course, there are differences in expression of this system according to the use that the organism made of the magnetoreception. For instance, I got at least 3 proposals rejected by reviewers that say, I do not perceive magnetic fields, so why we should study? That's great. So this is the actual situation of most of the reviewers. But of course, indeed, I believe there is some effect because I can prove it. So based on this concept of this MEGAR system, we propose that also plants may use the same system that migratory organisms are using, but probably in a different way and for different, of course, meaning, because plants are not moving. So they could use this system to adapt to situation more than exploiting to move toward other places. So we published this paper that was trying to demonstrate that also plants may have the same system. So first of all, we did this evolutionary relationship between the Drosophila melanogaster ISCA1 MEGAR gene, which is the one demonstrated by the Chinese group, and we showed that the ISCA1 is very much connected to most of the photosynthetic lower organisms, but also is connected to most of the plants. And the same is true for ISCA2, which is not as much involved as ISCA1 in magnetoreception, but is also connected to many, many organisms. As you can see, Drosophila melanogaster is quite linked to Homo sapiens. So this gene is also present in human. When we did the phylogenetic tree, we could see that actually Drosophila melanogaster and Arabidopsis thaliana were in the same clade for the ISCA1. But also it was interesting to see that you can see here Dictyostelium discoideum, Candida albicans, Chinorhabditis elegans, and Danioreo. So many organisms that are also organisms. And this is why now we are moving also toward these other organisms to see whether there is a relationship with this non-migratory relationship with this magnetosensor. So we start working on, let me tell you, real plants. So food plants, not only Arabidopsis, which is a fantastic mother plant, but nobody will ever eat it. So we thought it is important to concentrate on food plants. The first plant was lima bean, and we could demonstrate that there was a very nice effect on the hypomagnetic field or near magnetic field on the biology of this plant. So we did this experiment, and of course, once again, we were having control plants in the geomagnetic field and treatment plants in our system. So we analyzed leaf area, stomatal density, chloroplast ultrastructure, several biochemical parameters, including chlorophyll, carotenoid levels, and also, let's say, chlorophyll molecular changes, of course, also not only the determination of the total chlorophyll content, but also we focused on photosynthesis. So we checked the Rubisco activity and also the content, but also the activity of reaction center and the kinome reduction. What we saw, it was that even in this case, there was a clear interaction of the homologs of the MAGAR genes. In this case are Phaseolus lunatus ISCA2 and ISCA-A. And this was important because these two genes were completely responding to the lowering of the magnetic field. So we got an immediate response of the gene expression of these two genes, thinking that probably this system could also be involved in the magnetoreception in plants. I have several data that we are going to publish, and I will share with the Guy Foundation as soon as we publish these papers, because we have interesting results with mutants that we created on these iron sulfur cluster assemblies homologous genes. But what we saw, it was also that geomagnetic field induces a higher expression of gene involved in ROS production. So once again, if you take out the geomagnetic field, you have a lowering effect on ROS production. So in terms of structural analysis, we could see that there were changes in the ultrastructure of chloroplast. For example, also we did some metrics on the starch grains on plastoglobules, and they were changing according to the situation. But it was striking to see that the content of the rubisco was also lowered. So not only the enzyme activity, but also the content, and also this, which is one of the light harvesting complex in the photosynthetic system, was also lowered by the, let's say, the chronic exposure to low magnetic fields. In terms of photosynthesis, we could clearly demonstrate that there was a gap in the electron transfer chains of electrons between the two photosystems. This gap, we think it could be related to some quantum effects that happened in the radical pairs that are present in the photosystem too. But this very early event was affecting several things in the lima bean, like, for example, the production of ATP, the expression of the Benson cycle genes, carbohydrates production starts, and several other things. So there is something that is perceived very early and triggers a cascade of events that eventually give rise to lowering productivity and several other things. So another real plant was tomato. So in this case also, we'd analyze the behavior of these plants when you grow plants in the hypomagnetic field, meaning near null magnetic field. In this case, we were studying different developmental stages of the plant, from flower to fruit set. And once again, we could analyze two homologs of the MAGAR gene. And once again, we could see that they were responding to the difference in the geomagnetic field. We did an experiment involving gene expression, the evaluation, differential evaluation, gene expression of several, let's say, very important genes that are involved in the development. We were focusing on phytohormones and several phytohormones like auxin, gibberellin, cytokinins, abscisic acid, and the stress response like jasmonic acid and salicylic acid. And what we found at the end, there was first of all a clear response in the, let's say, in the quality of the fruits and in the quality of the tomatoes. So if you grow tomatoes in the low magnetic field, it changes the quality of the tomatoes in terms of antioxidant presence, lycopene presence, sugar presence, and so on. But also we could clearly see that there was some sort of correlation between the ISCA genes, the MAGAR genes, that are probably one of the first that perceive the changes in the geomagnetic field and then trigger gene expression that has effects on metabolism, so ROS production, pigments, polyphenols, proteins, phytohormones, and this also has an effect on development by affecting leaves, flowers, and fruit quality. So this was a nice experiment to be done on a real plant. Another plant that is often used in space simulation experiment is lettuce. Also in lettuce, there was an effect of hypomagnetic field, but this time we combined what we have already showed in the previous slide. So we combined the effect of lowering the magnetic field with lowering gravity. So what you see, this is the control plant. if you put plants inside the reducted geomagnetic field, the morphology is a little bit changed. But if you put the plants in the random positioning machine and you mimic zero gravity, this is the response of the plant. So roots are not anymore oriented as in the case of the georientation. And so the effect that you see is this one. So we did our RNA-seq analysis. If you compare the control plant in the geomagnetic field and you clusterize, let's say, the downregulated and the upregulated genes, you see immediately that the hypomagnetic field or the near-null magnetic field shows immediately differences in gene expression, and the same is true for the microgravity simulation. We could also see there were clusters of genes that were behaving in a similar way. For example, the cluster C, when you have downregulation in control, you have upregulation in both treatments, and the opposite happens with the cluster F, where you have upregulation in control and downregulation. This is interesting because it opens new avenues when you have to understand what it will be growing plant in a place that has low magnetic field and low gravity. So you know that there will be genes that will cooperate, and you will know that there will be genes that will also have opposite effects. So at the end, when we were doing this kind of study, at the end we could see that there were core adaptive mechanisms that were combining the effect of hypomagnetic field and the simulated gravity. In particular, you could see that if you lower the magnetic field, you have altered root architecture, reducing chlorophyll and modulating ROS. But if you reduce gravity, you have disrupted cellular architecture, photosynthetic efficiency, and then you have unique adaptive challenges. So at the end, what we could demonstrate is that Lectius as the way to adapt to this situation. We identified those genes that could be majorly involved in response to hypomagnetic field and to reduce gravity. And this is interesting because now we can target to those genes in order to adapt new plants when they will be transported to these extraterrestrial places, and we will know exactly what it will happen. Of course, we cannot reproduce radiation and several other nasty things that happen outside our planet, but these are two important points that we can further develop. One of the triggering of cell-to-cell communication in plants is the plasma membrane H+ ATPases, so the proton pump. The proton pump is important because it maintains the difference in the membrane potential, and we could find a correlation between a protein that is very much involved in the regulation of the cell metabolism, which is the 1433 protein. This is common in both animal and plants. And this protein actually in plants is able to bind to the plasma membrane and interact with the proton pump. So what we found is that in normal geomagnetic condition, there is an interaction of the 1433 protein that binds to plasma membrane and interacts with the proton pump. At interacting with the proton pump, it activates the autoregulatory ATP sensitive site of the pump, so it gets phosphorylated, and this creates the situation of pumping out the proton. Pumping out the proton creates the condition of the membrane potential in plants. So in normal conditions of the normal geomagnetic field, you have this kind of situation, so everything is balanced and the pump is working fine. But if you expose plants to near-null magnetic field, then you have an impairment in the binding of the 1433 protein to the plasma membrane. You have a reduction of the phosphorylation auto-activated site of the proton pump, and then you have a reduction of the activity itself of the proton pump, which is one of the causes of the late reaction that we can see in the homeostasis of the cell. Interesting, even in this case with maize, we could see that the Cry1 was interacting with one of the homologs of NIL-MAGR. In this case was the zeamycis Ca2. And it was also interesting to notice that melatonin was involved in this response to near-null magnetic field. If you combine melatonin with the clock that is present in plants, but also present and mostly present in animals, and you think about that melatonin reacts to magnetic fields, then you have to combine the jet lag to the place where you go, where the magnetic field is probably different. So this is interesting and opens a lot of questions about the role of melatonin and the involvement of melatonin in the magnetoreception. We actually know nothing about that, but we notice that melatonin is involved. So this was from this experiment the combining picture of the situation of roots and coleoptiles. Once again, there is a difference in the perception. It's interesting to note that the MAGR homolog genes, ISCA1, ISCA2, and the CRY1, CRY2, and Phi-A, Phi-B genes are also modulated, and as you can see, in opposite way, both in roots and cotyledon and coleoptids. And also there is a correlation between the near-null magnetic field and the geomagnetic field with regards hormones, ions, carbon and nitrogen metabolites and ROS. So definitely we can still confirm that even in maize, which is an important plant, there is an effect of perception of the geomagnetic field. When we talk about secondary metabolites, these are also important because we know that several plants produce bioactive compounds. So we published last year this paper by looking what was happening in a very famous secondary metabolite plant, which is peppermint. Peppermint produces essential oil through these glands, but the number of the glands was not affected by the near-null magnetic field. But when we were analyzing the essential oil composition, we noticed that there was a huge difference in the chemical composition. So we dissected the biochemical pathway of the menthol production by analyzing all the genes that are involved and of course during doing GCMS analysis to quantify the product of these genes. And we noticed that incredibly the content of the terpenoids is reduced, but the gene expression is increased. So there was something really difficult to understand. So the plants react to the magnetic field reduction by increasing the activity, but there is a bottleneck that regulates at the end probably the proteins that are produced by the transcription of these genes. So what we noticed was there was once again difference in the metabolomics, in the total protein context and in the total phenols. And also we could notice a difference in the photosynthetic activity of this plant. So what we thought it was that there was an effect very early, as we found in linapene, and this effect could be related to the quantum effect that happened in the radical pairs that are formed in the photosystem II centers. So what we think is that there are several players that are able to lower the photosystem II parameters, like reducing the photochemical quenching, but also modulating the non-photochemical quenching. But something happens at very, very fast timing. So I cannot disclose now the results, but we are doing time course experiment in 5-minute times. And we notice that there are huge difference in photosynthesis when you turn off or turn on the system. So nothing but quantum effect can react so fast. There is no time for reorganization of protein or degradation of systems or gene expression in 5 minutes. So we are quite convinced that there is something going on at the quantum level in the photosynthesis and particularly in the PS2 center that could be involved. What we notice in peppermint is that there is a strong upregulation of the production of ROS as the plant is perceiving a stress, But then there is a strong response of the detoxification routing. So there is a strong upregulation of the scavenging system, and the net result is the reduced steady state H2O2 production. So as you can see, the hypomagnetic field in peppermint are definitely doing something interesting at the photosynthetic level. So they interfere with the electron flux during the photosynthesis. They interfere with the ROS production, but this is paradoxical, as is paradoxical the increase in gene expression but decrease in terpenoid production. The paradox is that if you think about plants that react to stress with ROS increase, in the case of near null magnetic field, they react by decreasing ROS. This definitely has something to do with photosynthesis, has something to do with the modulation, even in this case, balanced by different genes, the magnetofield responsing genes. So the very last thing is this happened just 2 weeks ago. We published this paper where we combine 3 important factors. So we were mimicking Mars gravity, mimicking Mars magnetic field, and using the Mars regolite. So we were combining these 3 stressors. So using the system with the random positioning machine set up to 0.38 G, the geomagnetic field reduction, and we purchased this Mars Global Simulant and NGS1, which is an analog of the Mars regolite. Of course, we didn't trust too much the composition that was explained by the company, so we did a very deep geochemical analysis during this work, and we analyzed deeply this soil, this simulant soil, and we found many more important information. This was with a collaboration with the geochemistry department here in Turin. So they are very, very good in this kind of geochemical characterization. So based on this, we could actually understand that if you lower the gravity of Mars, you have some kind of response. If you lower the magnetic field of Mars, you have other response. But when you combine lowering gravity and lowering magnetic field, you have different effects. In case of lowering the gravity, the combined effect with the regolite is dramatic. So you have a very, very strong effect on the plant. And this is not the same in the case of the reduction of magnetic field. So at the end, what it concludes this paper is that when we will grow plants on Mars, the problem will not be the reduction of the magnetic field, will not be the reduction of the gravity, because plants still react to the gravity of Mars, but it will be the soil. The soil will be the major problem for growing plants in Mars. So actually what I showed you is something that connects everything to the quantum thread of the response of plants to geomagnetic fields. So I am deeply convinced that there is something quantum behind the plant responses that we saw. These are too fast, these are too reversible and too quick to claim for a particular receptor that has to be, for example, expressed and then can react. So there is something very simple, very, very basic that works on this. That's why we are focusing on iron. That's why we are focusing on something that is already responding to magnetism. And we definitely think that there is something going on in the photosynthetic apparatus that may trigger a series of events that happens later in the plant. So, of course, there is, from one side, we know to understand much more in order to optimize the response of plants to the geomagnetic field, but on the other side, we need to understand better what is the quantum environmental sensing system. We know radical pair mechanisms are the basic to understand quantum effects. And we know that there are so many radical pairs from mitochondrial to chloroplast to many other things that are interfering. Iron superclusters assembly are important because they provide iron and sulfur to the core centers where these radical pairs are working. So we definitely need to understand much more on this mechanism. I recently published a paper that summarizes some of these quantum response in case of stress. So I've been focusing on light, oxidative stress, and temperature stress, and try to identify which is the quantum effect that could be involved. And in case of quantum coherence, is clear for the light stress, where quantum tunneling appears to be much more involved in temperature stress. And the combination of the two, coherence and tunneling, is probably what dictates the response to oxidative stress. So what I showed you in this 1 hour, maybe more than 1 hour time. So we've been going through evolution, flowering, photoreceptors, mineral nutrition. We've been checking the difference between organs, focusing on the magnetoreceptor, whether or not this could be similar to what is happening in animals, focusing on photosynthesis, on proton pumps, and also the combination of microgravity in order to simulate what it will be in deep space, on Mars, on Moon. So, of course, we are also doing something else. We published a paper where we could demonstrate that ants lose their orientation, and this is because they have profound differences in the biomeans in their brains. We were analyzing the brains of the ants, and we noticed there were differences in the dopamine and serotonin. These induced behavioral changes, so we were testing also the ability of the ants to reconstruct riddles and to find the food. And once again, MEGAR and CRY was involved in this ant modulation. We have collaboration for studying Dictyostelium, Ceronaradictis, zebrafish. You should see zebrafish in solar system. They just align, they don't move. When you approach, they start moving, but they come back and they look like hypnotized in the absence of the geomagnetic field. And we have a project that has been approved, and because a group in research in Turin are exploring the possibility of improving the life of astronauts in space, because in microgravity there is a problem with the menstrual cycle. So we are growing uterus organoids to try to find the pharmacokinetics of the hormonal stimulation of the uterus. So everything is combining microgravity and also magnetic field. Finally, we got a grant to build the first ultimate machine to reproduce Mars conditions. So this machine, this is the size of the machine. We'll have, it's a very high-tech system. We have CO2 atmosphere at 7 to 9 millibar, the same pressure in Mars. We have -40 temperature. We will block the Earth magnetic field with a Helmholtz system. We will simulate the Mars-reduced gravity, and we will have different lighting system, including very strong UVC, B, and A light system made by LEDs. So, of course, we don't think that we will grow plant outside a greenhouse, but a greenhouse will stand to the condition of the planet, so reduce magnetic field, reduce gravity. Probably those greenhouses will have also reduced atmosphere, so we have to adapt plant to hypobaric condition, probably not 21% oxygen, probably 10% oxygen, more CO2 to make them happy, but we need to understand how they can react. We are working hard on producing overexpressing and KO mutants in order to understand how this plant can be prepared for this kind of mission. So there is a quantum to cosmic agricultural paradigm, which is to combine quantum physics to plant physiology and to space exploration in order to get the directed astrobiology. Of course, it cannot be done alone, these things. So I have to thank all the collaboration around Europe, particularly the Italian Space Agency, which is providing most of the grants that we have on this kind of space application. My groups here in Torino, in my group of plant physiologists, graduate students and PhD students, and some collaborators from the medical departments and biotechnology departments. Okay, thank you for your attention and greetings from Turin.